So, hello everyone. A very good evening to all of you. Please confirm if you can hear me and you can see the screen. Yes. Can I get some updates from you? Can I get some inputs from you? Hello Vijay. I hope so I'm audible and the screen is visible to all of you. So, in today's session, we have taken up Unit 6, that is plant physiology part. So, I'll be taking up the questions from your subunits 6C to 6H. Okay, but I'm not sure whether I'll be able to cover all the subunits or not, but still, no problem. Whatever is possible, we'll cover it today. Rest of the questions, we will definitely have a discussion in our next session. Okay. Yeah. So first tell me how many of you will prefer to go for unit 6 when it comes to CSIR NET exam? Usually what happens is that whenever we look into CSIR NET exam, there is a dilemma whether you go for unit 6 or unit 7. So are you all with unit 6 or you are also in between unit 6 and unit 7? Okay, I can see Sarita. Yes, you are going with unit 6. Okay. What about others? Hello, Rio. Unit 6, when it comes to CSI net point of view, remember it holds a very good percentage, very good weightage, and you can expect both B as well as questions from C as well. Okay, Varsha. So let's move on to the first question now. This is from nitrogen fixation, which is from subunit 6C. In the global nitrogen cycle, the following microbial organisms are involved in three important processes: denitrification, nitrification, nitrogen fixation. So the organisms' name are given here: Rhizobium, Nitrosomonas, Nitrobacter, Pseudomonas, Azotobacter. Now you have to identify correctly matched pair of process along with their causative agent. Have a look into the options given below and let me know the correct answer in the box. Okay, Joshna. So basically, when we say overall unit 6, nitrogen metabolism and stress physiology, it's not mandatory that you will always expect question from here. If you look into the pattern, like the previous year question papers, you'll find that they are being asked in a rotational basis. Like sometimes they will ask and sometimes they won't ask. But the thing is that even if they ask, it can be either part B or part C. They are not sticking to a particular section over here. So you can expect both. Yes. Can I get some answers for this question? So this is quite an easy question if you know the species and the nitrogen cycle as a whole. Yes, I can see Sarita has given the answer. What about others? Is there anyone else would like to give your answer? See, this is a very simple question. I guess everyone should answer this. Who are denitrifiers? Who are nitrogen fixers? Who are causing nitrification process? So every stage has their own unique microorganisms present. Okay, Joshna also come up with the answer. What about others? Is there anyone else would like to give an answer for this question? Okay, Vincent. Okay. Fine. That's really good to see that you all have given the correct answer. That is denitrification. It is caused by Pseudomonas. Nitrogen fixation is carried out by both Rhizobium and azotobacter, nitrification process can be carried out by nitrobacter. So let's check out the explanation now. The first thing is that you have to be clear with the nitrogen cycle. What is happening here? So nitrogen, you get it from the atmosphere. So either physical, chemical or biological method, they get fixed. So lightning, they can cause fixation, or maybe with the help of nitrogen fixing bacteria, it will be fixed, which converts the absorbed nitrogen into ammonia. This ammonia will be given to nitrifying bacteria so that it converts into the assimilatory form, that is your nitrate. And after the assimilation, whatever the remaining nitrogen is there, they has to be recycled back to the atmosphere. So this is carried out by your denitrifying bacteria. Meanwhile, you will also find ammonification process also going on. That means directly ammonium ions gets converted into nitrite, which in the presence of a nitrifying bacteria can be converted to nitrate and they can be assimilated. So basically ammonia is also used directly by the plants. How? By the conversion into amino acids. And nitrate is directly utilized. So if you see the steps involved over here, nitrogen fixation involves conversion of nitrogen to ammonia, which is being carried out by nitrogen fixers we call as a diazotropes. Then ammonia gets converted to nitrite by two bacteria, that is Nitrosomonas and Nitrococcus. Nitrite is again converted to nitrate because, as I told you, this is the assimilatory form. Nitrobacter and nitrocyst. And finally, assimilation, then denitrification carried out by the decomposers known as Pseudomonas. Now, when it comes to biological fixation, you have got two categories of that. First category is symbiotic fixation and the second category is free-leaving fixation. Symbiotic fixation means what? They require a host to carry out the process. Free-leaving means they have the inner ability to carry out the fixation. So if you see all the rhizobial species, that is Azorhizobium, Bradyrhizobium, Meso, Rhizo, Cyno, everyone for the host plant is the leguminous. It could be legumes or parasponia. Actinorhizal, Frankia. Ganera, nostoc. Azolla, anabina. Sugarcane, acetobacter. Miscanthus, azospiralium. Now, if you look into the free-living category, you have cyanobacteria, aerobic bacteria, facultative, and anaerobic. Cyanobacteria, we have Anabina, Calothrix, and Nostoc. Aerobic, we have Azospiralium, Azotobacter. Facultative, that means they can survive in both the conditions. We have Bacillus and Klebsiella. Then we have non-photosynthetic, Clostridium, Methanococcus. Photosynthetic, Chromatium, and Rhodospiralium. So this table is important from examination point of view. Once upon a time, they used to ask a lot of questions from this particular table, specifically in the form of match the following. Okay, so be clear with this. And here I just wanted to mention few microorganisms uniqueness. That means check out nostoc. It is present as a symbiotic fixer. It is also present as a free-living fixer. Similarly, we have anabina. They are also present as a nitrogen fixer, symbiotic and free-living. Then we have acetobacter. So remember not to be confused with this term. Acetobacter, azotobacter. Just a difference of few letters. Aceto comes under the category of symbiotic fixation. Azoto comes under the category of free-living. Okay. So for Azotobacter, that comes under the category of aerobic organism. Okay, so these are the few things which you can have a note on. Including your nostoc and anabina, I missed out one more organism. Azospirillum can also do the same, symbiotic as well as free living. Okay, so just have a look into this table. As I told you, important. Let's have a look into our second question. Following are certain statements regarding nitrogen uptake and assimilation by plants. Plant roots take up nitrogen in the form of nitrate or ammonia. Ammonia take up plants can be directly assimilated into amino acid. Amino acids are synthesized exclusively in plastid and chloroplast of root and leaves. Nitrates can be stored in vacuoles of both roots and leaves. Just identify the correct combination from here. Identify the correct combination of statements from here. Okay, I can see Sarita has already given the answer. What about others? Yes, Varsha, Vijay, Elna. Any inputs from your side? So basically it is related to what? Assimilation process only. Yes, Pavan, do you have an answer for this question? Very good evening to you. Apart from Sarita, is there any other answer from anyone else? See, this is also quite similar to what I had given you previously, wherein I have mentioned some details which you can correlate to this question as well. So, okay, Varsha. So you have given 1 as the answer. Sarita has mentioned 3 as the answer. So what I understood from both of your answer is that you are quite confident with AB. The third one is confused whether C should be the answer or D should be the answer, right? Okay, no problem. Anyone else? Anyone else would like to add your answer here? Okay, let's find out what is the correct answer is. Okay, Elna, it is 3. Okay. And yes, you all are absolutely correct. It is A, B, and D. So why the statement C is incorrect? Do you have an answer for that? Why the statement C is incorrect? NO3 excess and NO2 is toxic, so cannot be stored in vacuoles. Yes. Yeah. So let's have a look onto that. See, many amino acids, particularly those, so why we are finding out why the statement C is incorrect, because what we got A, B, and D as correct. Right? Just check it out now. Many amino acids, particularly those that are involved in primary metabolism, they are synthesized in plastid. Now that includes glutamate, glutamine, aspartate, and asparagine. So you all might be aware of glutamine synthetase and glutamate synthase pathway, which is one of the important pathway when it comes to nitrogen metabolism. We also call it as GS-GOGAT pathway. So what is their role? They play a key role in nitrogen assimilation by converting these ammonium ions into glutamine and glutamate, which act as an amino group donors for the production of other amino acid. Then chloroplast and leaves are also involved in synthesizing amino acids that is linked to photorespiration. So it could be glycine, serine, right? Next, some amino acids are also synthesized in the cytoplasm as well. If you have studied secondary metabolites, you might be aware of shikimic acid pathway. Shikimic acid pathway is responsible for producing aromatic amino acids. So these are occurring in plastid and also involves cytoplasmic steps as well. Apart from all these things, certain amino acids are also involved in mitochondrial metabolism. That means mitochondria also produces few. Then adding to that, TCA cycle. They also provide precursors for amino acid biosynthesis in mitochondria. So that's the reason statement C is incorrect. I hope so you got it, Varsha, why the statement C is incorrect. This is an image that represents the summary of how the nitrogen assimilation takes place. As you can see, we have got the nitrate through a kind of a symporter enters inside the cytoplasm. Upon converting to nitrite, can be released into the chloroplast. Then with the help of GS go-get mechanism, glutamate is synthesized. Where it is synthesized? In the chloroplast. After this glutamate synthesis, they will be released into the cytoplasm, where it gets converted to other amino acids like aspartate, asparagine, and others. So this is one proof that it is not exclusive for plastid. Then I mentioned about the TCA cycle. So this is your TCA cycle, right? So you can see oxaloacetate gives rise to aspartate, asparagine. 2-oxoglutarate gives rise to glutamate. And from there you get other amino acids. Okay. Apart from that, glucose metabolism, alanine, which is obtained from pyruvate, 3-phosphoglycerate gives rise to serine and other 2 amino acid. PEP, they also give rise to aromatic amino acid. So that's the reason the statement C is incorrect over here. Moving on to our next question. This is a match the following plant hormones with their receptors and the response. Auxin and gibberellins, cytokinin and racinosteroid, transmembrane and soluble receptor, response mediated by phosphorylation or dephosphorylation, or proteasome mediated protein degradation. Find out. Plant hormones is an important topic when it comes to CSIR NET point of view. You cannot even think of avoiding it. You get it in the form of match the following, or you can expect it in the form of statement-based questions. So never ever skip these hormones. So we have 5 major hormones: auxin, gibberellin, cytokinin, racinosteroid. In fact, more than racinosteroid, we have ethylene and abscisic acid. The 5 major hormones. Try to be clear with their synthesis part, their signaling part, their functional aspect, and apart from that, the combination hormones. That means auxin, gibberellin, what they do, auxin, cytokinin, what they do, abscisic acid, gibberellin, what they do. So those kind of combination hormones also based questions do come in the exam. So have a look and let me know what is the correct match over here. Okay, Sarita, so you got option 2 as the answer. Okay, what about others? So when you try to solve a question, always try to identify what is the reason behind the answer for that question. Like for match the following, you don't have any such reason and all. But like previous question and all, when we were discussing, you have to be clear why the statement A, B, and D is correct. But at the same time, you should also analyze why the statement C is incorrect. So if you do so, if you make a habit of doing so, you will find that you feel confident with the concepts which you hold on. Okay. Yes, can I get some more answers for this question? Is it tough? Plant hormones. So initially when I asked who will go for unit 6, I can see many of you has said, but very less answers I can see. Try out, everyone. See, this is not your final exam, right? You are trying out something. So with your knowledge, if you are giving the correct answer, be confident. And if you are choosing a wrong answer, try to learn from it. So what should be the correct answer from here? Okay, beautiful colors. It's been a long time. Got an answer from you. Okay. Let's check out what is the correct answer for this question. The correct answer for the okay Kalki. So I got different answers 2, 4 and 3, but the correct answer over here is 2. Now why 2 is the answer? So what does 2 indicates? AB2. Auxin and gibberellins, these are soluble kind of receptors, and their response is mediated by proteasome mediated protein degradation. BA1, cytokinin and brasinosteroids, they have transmembrane receptor whose response is mediated by phosphorylation and dephosphorylation event. You have to be very clear with the signaling pathways of each of this plant hormone. So I have an image for you, as you can see over here. We have 5 major plant hormones I told you. Among them, you can see auxin, jasmonic acid. So again, that is a defense response hormone. Gibberellic acid, everything you will find as soluble and present in the nucleus. Right? Then cytokinin and ethylene, you will find it in the ER. These are the examples of different receptors. So it's not necessary that every hormone will have one receptor. It can be many. Similarly, abscisic acid, you see, they have cytoplasmic receptors as well as membrane-bound receptors as well. Brasinosteroids, if you check out, you will have it is bound to the cell surface or the membrane. So, depending upon the location, you have got the receptors located at that position. Okay. So, looking on to the signaling pathway. So, do you want me to explain the signaling pathway or shall I move to the next question? Just let me know. Do you want me to explain this signaling pathway or you are aware of this? I have signaling pathway for auxin, cytokinin, gibberellin and brasinosteroid. Shall I proceed with the explanation or will you take, do you have idea about it? Yes? See, basically, whenever you look into the any hormonal signaling pathway, remember that you should remember two things. One is what is their repressor, and second thing, who is their transcription factor. And of course, where is the receptor and where is the ligand where is the receptor located? That is also important. Okay, so I'm just taking up all the hormones, all the means whatever was given in the question. So what happens is that in the absence of auxin, so IAA indicates auxin, right? So in the absence of auxin, we have got the repressor known as OX-IAA binds to it. So we call it as an heterodimer because two different molecules are bound. So in that case, there is no gene transcription taking place, right? Now, when your auxin goes and binds to the corresponding receptor, what happens is that your OXIAA, that is considered to be a repressor, it will be targeted for proteasomal mediated degradation. So now what happens? Your transcription factor ARF is free, which will undergo dimerization known as homodimer. And they will go and sit onto the specific response elements present in the DNA. So every protein will go and bind to the DNA only to their specific sequence. So here also you have got a palindromic auxin response element. Palindromic means they are mirror images to each other. After that, what happens? Transcription begins, and whatever necessary is there for auxin response, whatever the function is there. So depending on that, that genes will be synthesized and the function will be accelerated. Now, in this image, you can see that this repressor itself will serve as an early response gene. So it has got a dual function. If auxin is not there, it serves as a repressor. If auxin is present, they will carry out the early gene response. So that is your auxin mediated pathway. Now, if you look into the gibberellin mediated pathway, gibberellin is somewhere related to your GPCR pathway. Okay. So now what happens here is your gibberellic acid can go and bind to their corresponding receptor, and your G protein will be activated. Now, this can activate two pathways. First one is calcium dependent pathway. So, calcium dependent pathway will directly break down the starch molecules and they will release the food to the embryo. So, there is no specific steps involved. But calcium independent pathway, what it will do? Once your GA signaling the hormone is activated, they will enter inside the nucleus. Okay. Once they are inside the nucleus, here who is the repressor? DELA is the repressor. So they will target the DELA for degradation. So now the DELA repressor is removed from here. GAMIP gene, which is considered to be the transcription factor here. So that gene will be free. And they will carry out the mRNA synthesis, which will go outside, serves as a transcription factor producing the protein. Now, this protein will go and bind to the promoter region of alpha amylase gene. And they will start synthesizing mRNA and finally the necessary proteins. And what is the function of the amylase over here? They will be targeting the polysaccharides to degrade them into monomeric units, because the embryo can take up only the monomeric form of food, not the polysaccharides. So they will break down the starch and convert them into monomers so that the endosperm or the embryo can consume it. Okay, so that is gibberellin signaling pathway. Third one is cytokinin signaling pathway. So cytokinin, they will go and bind to their corresponding receptor. Within the receptor, there is a domain known as chase domain. So you all might be aware that cytokinin and ethylene are the two plant hormones which serves as a two-component system like bacteria. That means they will have a histidine kinase domain, they will have a receiver domain, right? So this cytokinin goes and binds to the chase domain of the receptor, which in turn activates the histidine kinase domain first. Upon ATP hydrolysis, they will transfer the phosphate to the receiver domain. And who is there in the receiver domain? Aspartate is present. So it will transfer the phosphate to aspartate here. From here, the phosphate will be transferred to another protein known as AHP. Now what happens? This AHP, upon phosphorylation, which is present in the cytoplasm, releases into the nucleus. And what they will do? They will transfer the phosphate to two receptors over here or two proteins over here. That is type B ARR and type A ARR. Type A ARR is the main protein that is responsible for carrying out cytokinin response. So what does this type B phosphorylated version will do? They will accelerate the activation of type A RR and cytokinin response will be generated. So once the enough of response we get, this type ARR will give it serves as a negative regulator. So it will block the AHP. So there won't be any response. And at that time, this type B will be degraded by protease. So that is how your cytokinin signaling works. All the three signaling is clear? All the three signaling pathway is clear to all of you? Yes, Sarita, Krishna, Henry. Is it clear? Okay. And the last one is the brasinosteroid. From here, you get very less questions, not frequently, but still you can have a look onto that. So this is a receptor for your brasinosteroid, that is BRI1, which is a kind of a homodimer. And the kinase inhibitor is there, which will inhibit the receptor in the absence of brasinosteroid. Now, along with that, another protein, BAC1, is also there. Now, what happens is that whenever the brasinolide, that is the active form of brasinosteroid, goes and binds to the receptor, what happens? The kinase inhibitor will be released. So, when the kinase inhibitor is released, the receptor gets activated. So, your BKI1 and BAC1 will undergo transphosphorylation. Now, they will activate a kinase known as BSK. This kinase will go and activate a phosphatase, BSU1. This phosphatase, so what is the role of a phosphatase? To remove the phosphate from the target. And who is the target over here? BIN2 protein. So two things you can remember over here. If BIN2 is present in the phosphorylated form, they can enter inside the nucleus and phosphorylate the transcription factors. Upon phosphorylation, the transcription factors can be either degraded in the nucleus or they will be released into the cytoplasm and sequestered over there. So that is when your BIN2 is phosphorylated. If BIN2 phosphorylation, if we are removing the phosphate group from here, BIN2 will be dephosphorylated. So this dephosphorylated BIN2 is degraded by proteasome system. So at that junction, what happens? Your transcription factors are free. So when your transcription factors are free, they are responsible for carrying out the gene expression. Okay? So that is how all the four signaling pathways work. But remember, you also need to study ethylene as well as abscisic acid as well. They are also equally important. Okay? So this is just a reminder that we have Lakshya batch going on and you can always enroll into this batch so that you will get a free internship program, that is molecular biology techniques. And through this batch, you can access unlimited classes, 24/7 chat support. You will have exclusive study portal. Then some smart tools will be there that will help you in revision, that is Conceptica series. Weekly, monthly quest will be available. If you have any queries, just you can talk through this number or you can visit stores.biotecnika.org. Okay. Moving on to our next question. So in this question, the plant hormones, auxins and cytokinin and their interaction plays a very important role in regulating apical dominance. Following figure represents an experiment that is related to the study of gene interaction that influence axillary bud outgrowth of dormancy. So in the image, you can see QZM is being represented over here. Now, according to the statement, I mean, according to the figure, some statements are also provided, where you can see X is an auxin, which maintains the expression of Q and Z, repressing the M. Y is a cytokinin, promoting axillary bud and is induced by M. Decapitation activates X. X is a cytokinin that represses M. Now you have to identify the correct combination of statements from the given image. Identify. So as I told you, you can expect combination hormones based questions in the exam. And one good thing about Unit 6 is that, unlike other units where you will get questions from outside, from different reference book, for plant you have got only one book, that is your Tays and Ziegler. And majority of times you get questions from this book only, directly from this book. In fact, they directly take the images also from the book itself. And this is one such example of that. Identify. So first here you need to find out what does Q, X, Z and M stands for. XY is given, Q, Z, M is given. So you have to identify what are they, and then you can get into an answer. This is an intact plant. This is after decapitation what happens. This is your axillary bud growth. So somewhere they are representing the functional aspect of these hormones only. Sarita, I did not get an answer from you. What happened? Usually I get your answer first. And others, try out. It's not tough if you understand what does this letters indicate over here. Yes. No one is able to answer. Do you know what is an apical dominance is and how this auxin is related to that, how cytokinin is related to that? Do you know that? Okay, I got an answer from Sarita mentioning A and B. Okay, yeah, you can mention, you can take this as A, B, C, D, okay? By mistakenly, I have mentioned here as letters, but here numbers. Yeah, there is a small mistake. You can consider these statements as A, B, C, D. Instead of 1, 2, 3, 4, you can consider it as A, B, C, and D. Yes, okay, Sarita. So good to hear that you are providing explanation to your understanding according to the question. That's good. Others, try it out and check if A and B are correct or if you find any other option as the correct answer. So which book you all follow for plant physiology, everyone? Which book do you follow for Unit 6 from CSIR NET syllabus? Meanwhile, others can give the answer. It's not that tough, everyone. Okay, let me show you the answer then. The correct answer is what you mentioned is correct, Sarita. It is A and B. And others, have a look over here. See, This image is directly taken from your Tays and Ziegler book. And it is just one example I have shown you. Like this, several examples are there which is image based question, and it is taken directly from the book itself. Cytokinin will modify apical dominance, promoting lateral bud growth. So remember, apical dominance, as mentioned by Sarita, is carried out by your auxin, whereas the lateral bud growth is carried out by cytokinin. So here, IPT gene is off, cytokinin oxidase gene is on. IPT is responsible for producing cytokinin. So when you have got an intact shoot apex, your auxin can move. Okay. So at that stage, we don't require cytokinin, and whatever the cytokinin is there, it has to be degraded. So we have cytokinin oxidase on. Now, if you decapitate it, that means if you remove the top portion, you know that apical dominance means the top portion only. So if you decapitate it, slowly cytokinin production increases. So obviously IPT gene will be on and the oxidase enzyme will be off. But what does the cytokinin will do? Once they reach a particular size, they will induce the activation of auxin. So again what happens? IPT will be off and CKX will be on. That is going back to the first one. So exactly the same image is shown over here also. Right? You are able to identify what happens in the intact plant and what happens after decapitation and what is the axillary bud growth. Right? I hope so it is clear to all of you. Yes? Am I clear to all of you? The question is clear to all of you? Let's move on to our next question then. Here are some statements given in relation to seed maturation. Seed maturation involves mainly the accumulation of storage products like starch, lipids, and proteins. A large number of chaperones, including the family of Lea proteins, plays a crucial role in the desiccation process of seeds. Moisture content gets reduced with the maturation of seed. ABA and gibberellin both can promote seed dormancy. Identify the correct combination of statements from here. So first I have a small question to all of you. Can you please tell me which hormone is responsible for seed maturation? So very quick response, Sarita. Good. So please tell me everyone which hormone is responsible for carrying out seed maturation. Seed maturation I am asking. Which hormone is responsible for carrying out seed maturation? No, ethylene is for fruit ripening, right, Sarita? The major function of ethylene is fruit ripening, right? Then do you think it will induce maturation as well? What about others? Yes, you are correct, Sandhya. Seed maturation is induced by abscisic acid. What do you mean by seed maturation? Seed maturation means you are giving a time period for a seed to develop some genes so that they can synthesize the necessary genes that will be helpful for them to survive in the extreme conditions outside. Extreme conditions means the drought condition, flood condition, or extreme temperature, or maybe like hot or cold temperature. So based on that, we can see many plants are surviving, but many are not. Why? Because they have specific genes being synthesized in them, and that is promoted by your abscisic acid. Now tell me the correct answer for this question. Yes, apart from Sarita, is there anyone else would like to give your answer? Sandhya, would you like to answer this question? Okay, Sandhya also opted for option 4. Anyone else? Anyone else would like to give the answer for this question? Okay, Elna. So previous questions you did not answer. Elna, what happened? You were not aware of the answer? Okay, let's find out the answer for this question. The answer for this question is what you all have said. Absolutely correct. 4. Now, can you tell me why the statement D is incorrect? Why the statement D is incorrect? Abscisic acid and gibberellin both promote seed dormancy. Why this statement is incorrect? Why the statement is incorrect over here? Rest of the things okay, it's fine. Gibberellins, yeah. Yes, you are correct. So, see, during seed maturation, storage products such as starch, lipids, and proteins accumulate in the seed to provide energy and building blocks for germination. This phase is crucial for developing seed viability and long term storage capability. That's why statement A is correct. If you remember, I told you that there are some genes which needs to be expressed at the time of maturation. So, one such example is the LIA proteins. These are a group of chaperones which will protect the cellular structure during the desiccation process of seeds. They prevent protein aggregation and help maintain the membrane stability even under low condition, ensuring the seed viability. So that is also correct. Third statement, as the seeds mature, their content decreases significantly. So most of the seeds, if you see, they dry out to about 5 to 15% of moisture content so that they can survive in a dormant state. Dormant means it's an inactive state. So that's why statement C is also correct. Now, why statement D is correct? As you mentioned, yes. Abscisic acid is the one which promotes the seed dormancy. Whereas gibberellins are the one which promotes seed germination by breaking the seed dormancy. So that's the reason statement D is incorrect over here. I hope so, Sarita, you got it? The difference between maturation, germination, dormancy and all? You got confused while giving the answer for seed maturation? Right? I hope so everyone got that clarity. Which hormone is responsible for what? Okay. Moving on to our next question. Following are certain statements regarding phytochrome interacting factors, a family of proteins that regulates the photomorphogenic response in plants. PIF promotes scotomorphogenesis by serving as a transcriptional activator of dark-induced genes. PIF on interaction with PR gets phosphorylated followed by degradation via proteasome complex. Degradation of PIF takes place in the presence of light. PIF induced genes are not expressed in light. Identify the correct combination of statement from here. So this is the part of your sensory photobiology. So sensory photobiology also a very good topic for CSIR because they ask so many questions from this topic. And most of the times they ask in the form of part C, statement based questions. So don't forget to involve or include this topic while you are studying. Yes, got an answer from Sarita. What about others? Is there any other answer from your side? Sarita gave 2 as the answer. Any justification for that, Sarita? So I can see throughout my sessions how many sessions I have taken so far. I can see you have really interacted very well with us, and it's highly appreciative. And I am expecting that your revision is going very good for the upcoming CSIR NET exam. Okay, Sandhya, then if you have not revised it, I will definitely suggest you that never skip this topic and make sure that you revise this topic without fail. Okay, so any answers, any other answer apart from Sandhya and Sarita? Anyone else would like to give any answer for this question? Okay, let's check out the answer now. The correct answer over here is 2. That is A, C, and D. So, why the statement D is incorrect? PIF on interaction with PR get phosphorylated followed by degradation via proteasome complex. Yes, Joshna, you are also correct. After a long time you came. Initially you mentioned it, but then you left out, I guess. Yes, Purnima, yes, correct. So statement A, C, and D are correct. How? For that, you have to understand this image properly. So, what does this image indicates? 2 conditions. First condition is dark phase. Second condition is light phase. So, this is integration of light and GA signaling in Arabidopsis plant. So, in the dark phase, you will always find gibberellic acid is present in higher concentration. So, if that is present in higher concentration, obviously you can expect long hypocotyl. So, you have phytochromes. Phytochromes in the dark, they are inactive. So, they exist in PR form. So, gibberellic acid will enter inside the nucleus, binds to their corresponding receptor GID1. Then they will add ubiquitin tags to the GRAS domain of DELA protein because DELA is their repressor. So F-box element will add the tags and they will discard that into the dustbin. So who is the dustbin here? 26S proteasome mediated degradation. So because of this removal, what happens? Your PIF is free. Phytochrome interacting factor is free. So they will go and bind to their corresponding G-box elements present in the DNA via a basic helix loop helix. Upon binding, they will carry out the gene expression required for whatever is their requirement, that is specifically for hypocotyl elongation. Unlike that dark phase, in the light phase, what happens? We have low GA. So, obviously, short hypocotyl will be formed at that time. But remember, it is in the light, your phytochrome is present in the active version. That is, your PFR form is present. So, what happens is that this PFR enter inside the nucleus and target the PIF for degradation. So, what happens? Your transcription factor is degraded now. And also the repressors are active here. So repressors also will target the PIF for degradation. So when transcription factors are undergoing degradation, what happens is that there won't be any gene expression. So that's the reason, remember, in the dark phase, gibberellic acid will be more, hypocotyl elongation will be high. Whereas in the light phase, gibberellic acid will be low and hypocotyl elongation will be short. Okay. Now, if you look into this image in a different version, in case if you confuse over there. In the dark condition, your corresponding gibberellic acid will go and bind to their corresponding receptor and they will target their corresponding repressors to proteasomal mediated degradation. So, depending upon the requirement, they will carry out the gene expression. But when light is present, I told you phytochrome is active. So, phytochromes will enter inside and they will target the PIF for degradation. Della will also do the same. So, what happens in the light? PHY will stimulate the degradation of PIF. Della binds to the BIF, both of which prevent the expression of expansion genes, that is, elongation genes, we can say. Okay. So, you can understand through this image, or you can also understand through this image, completely your choice. Moving on to our next question. Directional growth of the plants is induced by the light called phototropism. So, some statements on phototropism are given below. Phototropism is a photomorphogenic response. FOT1 and FOT2 genes will mediate phototropism. Cry1 and Cry2 genes although help to perceive blue light but are not involved in phototropism. Perception of blue light by phytochrome A photoreceptor initiates phototropism. Identify the correct combination of statements among the following. So again, another question from sensory photobiology. See, among all the subunits, right from A to H, right? In A to H, what happens? A is important, B is important. That is regarding photosynthesis, photorespiration and all is important. Apart from that, nitrogen metabolism and stress physiology, I told you, they are always asked in rotational basis. Rest, your plant hormones, sensory photobiology, and secondary metabolites. They are also equally important like your photosynthesis. Second level of important topic is solute transport. After these three topics, you should shift over to solute transport. So they can ask one question, they may not ask one question like that. Yeah, they can ask even B questions from part B as well for solute transport. Secondary metabolites is also important. Like your plant hormones, there also you can expect match the following kind of questions the most. Okay, Sandhya. Sandhya has mentioned phytochrome A can be sensitive to light. Okay. Okay, Purnima. So you had mentioned A. A in the sense option 1 you meant, Purnima? Or the statement A. A is there in almost all the statements, like all the combinations. Okay, 1. Fine. So Sarita, Sandhya, Purnima has given 1 as the answer. Any other answer? Kalki has given 4 as the answer, A, B and D. Okay. Any other answer? That is also important, Sarita. Plant defense means the secondary metabolites. That is important from examination point of view. That's what I said you. Photosynthesis, respiration, plant hormones, sensory photobiology, and plant secondary metabolites. All these topics are equally important. Followed to that solute transport, then you have nitrogen metabolism and stress physiology. Still you have time, Sarita. You can prepare for that. Okay, then moving on to our answers now. So the correct answer, as you have mentioned by all, that is first. Statement A, B and C is correct. So can anyone tell me why the statement D is incorrect? Purnima, Sarita. So Sandhya has given some link to the answer. Purnima and Sarita, any inputs from your side? Yes, Joshna, any inputs from your side? Why D is incorrect? Kalki, I hope so you are able to understand. You meant D is correct. Okay, I'll come to the explanation part shortly. What about anyone who knows the explanation? Okay, see, check it out. Phytochrome A is not a blue light photoreceptor. Instead, it is sensitive to red and far red light, not the blue light. Phototropism means it is the growth response of the plant towards a light source, initiated by blue light perception, and these are carried out by a class of photoreceptors known as cryptochromes and phototropins. So we have two types of photoreceptors in plants. One is the phytochromes, which is red and far red light. Second one is the blue photoreceptor, which is blue light. They have cryptochromes and phototropins. Now remember, at the time of low intensity, chloroplasts try to accumulate at the upper region. So we call it as a chloroplast accumulation response carried out by FOT1. Similarly, when there is a high light intensity, it can cause organelle damage. So all the chloroplast will translocate to the side region. So we call it as a chloroplast avoidance response, and this is carried out by FOT2. Another part is that cryptochromes is of two types, Cry1 and Cry2. Cry1 is stable in light, whereas Cry2 is rapidly degraded in light. Both of them are blue light photoreceptors, but they mainly regulate seedling development, circadian rhythm, and flowering rather than phototropism. They perceive blue light, but they do not play a direct role in phototropism. Okay, so that is the reason. I hope so you understood the aspects of different topic. I know we are left out with solute transport, secondary metabolites, stress physiology, but no problem. As I told you, in my next session, we will be covering the rest of the topics. Okay. Thank you so much, everyone. See you again in the next session and expecting these kind of interaction in the upcoming times as well. Thank you.